Hi, my name is Sangeeta Bhatia. Today I'm going to be telling you about tissue engineering. In particular, I'll be telling you about engineering tissue replacements for patients. In this seminal paper in 1993, Bob Langer and Jay Vacanti described the concept of tissue engineering. The idea was that one would take a degradable biomaterial, basically a plastic, and mix cells with this degradable biomaterial, culture them together, and create an engineered tissue. These hybrid tissues that are part cell and part material could then be implanted for patients with tissue failure of various kinds. In this same paper, they examined the number of procedures per year that were done for patients that had tissue failure of some sort. And you can see that many, many millions of procedures are done for all kinds of medical conditions every year. So, how has this gone? Well, first, one of the challenges. If one looks at how tissues are organized in the human body, we see that they're hierarchically organized. So, for example, here is an image of the liver. And if you zoom in on the liver, you can see this fundamental unit of the liver, which is known as the liver acinus. This unit is about 1 millimeter in diameter, and there are many millions of these units in the liver. If one zooms in on these further, one can see that within this acinar unit are hepatic cords. And these hepatic cords are rows of hepatocytes, liver cells, that are organized in a way to process the blood efficiently as it's flowing through the liver. Finally, if one zooms in all the way to the single cell level, one can see that these hepatocytes, these liver cells, are surrounded by at least 5 other cell types in their home, in their what we call microenvironment. So these are the challenges of the tissue engineer, to recreate the microenvironment of these cells in a way that there could be many, many of them in healthy microenvironments in tissues that could replace the tissue function of a damaged tissue. So, first you might ask why one would create a bioengineered tissue that's part plastic and part cells. This is a very famous image of one of the first tissue engineered pieces of cartilage in the shape of a human ear in the back of an immune-compromised mouse. So why would one do this? Well, tissue engineers would typically do this only if one needed to improve upon one of the following existing medical therapies. If no simple medical device could improve the tissue function, for example, an implantable hip is a piece of metal or a piece of plastic, and if that could achieve the tissue function that you were interested in, you wouldn't create a hybrid device. A drug, if one could administer a drug to a patient, again, one wouldn't need a hybrid device. A cell that was minimally manipulated. This is the term that's been invented by the United States Federal Drug Administration, and it refers to cells that come out of the body and are reinfused with very minimal manipulation. So if one could minimally manipulate a cell, one wouldn't need to engineer a tissue to improve the tissue function. A surgical reconstruction. If one could simply borrow another part of the patient's body for replacement of the tissue that's damaged surgically, again, one wouldn't need a complex function replacement. And finally, a transplant. If one could use a cadaveric or donor organ, a whole organ, and replace the tissue function of interest, one wouldn't need a bioengineered tissue. But importantly, these criteria are often still not met, and one does need to, in fact, turn to tissue engineering. So there's actually a spectrum of tissues that can be engineered, and all of these fall in that category. So engineered tissues include biomaterials and cells in various combinations. And on the first slide, I described to you biomaterials that house cells. So these are plastic typically and cells together in a hybrid form. But there are also various incarnations of these engineered tissues. So for example, there are complex biomaterials that can be implanted and recruit cells. So upon implantation, they don't carry any cells, but they can recruit host cells and become hybrid in situ. These are also in the category of what one would call engineered tissues. And finally, there are materials that are completely derived from cells. So we know that cells themselves can make scaffolding, which is called extracellular matrix. And if one cultures cells in the laboratory and creates structures that are part cell and part extracellular matrix, those also can count as engineered tissues, but they have no synthetic materials within them. So all three of these categories of engineered tissues have been widely experimented on with varying degrees of success. And what I'd like to do now is to show you some examples of each of these. So we're going to start with acellular biomaterials. This is an acellular biomaterial, which is a very effective engineered tissue. It's called small intestine submucosa. It has no cells whatsoever. So if we look in this bottom image here, what we see is a cross-section of the intestine, in this case of a pig. So we see these villous structures of the intestinal lining, and that's how the intestine absorbs nutrients. And underneath that structure are these muscular layers. And lying between these two layers is what's called the submucosa. The submucosa looks like this on an electron micrograph. And this is a very strong extracellular matrix structure made up primarily of collagen and mixed with lots of other extracellular matrix types of molecules. So what was done originally by Steve Badylak and co-workers and many other groups since is that one can take porcine small intestine and strip away the cells with various detergent type procedures and come up with large scaffolds that are acellular that are very strong mechanically. It turns out that when one implants these at sites where regeneration is required, so for example in tendon or ligament repair, these recruit host cells by inducing proliferation and differentiation of those cells and promote wound healing very effectively. These types of materials have been in millions and millions of patients. Another more complex example is a hybrid example. So I told you before that one can combine biomaterials and cells to make hybrid engineered tissues, and this is one of the best examples, successful examples of having done this. So here we see engineered skin. So on this side we see a cross-sectional micrograph of what human skin actually looks like, and we see that there's a dermal layer here and an epidermal layer here. The dermis is composed of connective tissue cells like dermal fibroblasts, and the epidermal layer is composed of epithelial cells, which undergo what we call stratification as they mature and form this multi-layer surface. And way out here is where the skin interfaces with air. So this structure is what's damaged in the case of burn, for example. And tissue engineers sought to create tissue-engineered constructs to replace this skin. So on this side, what you see is an engineered version of skin. And in this material, what's been done is a biomaterial, a collagen gel, an extracellular matrix gel that has the consistency sort of like Jell-O, has been mixed with dermal fibroblasts. These dermal fibroblasts remodel the collagen, and they form a very tight meshwork. On top of that meshwork, one can see those epithelial cells, the epidermal cells, and they will stratify and form a multi-layer structure on top of that dermal layer. And this engineered skin can be implanted in the setting of burn or other injured skin and promote wound healing and to a great benefit. Another example of a hybrid tissue engineered system is this one. This one was developed originally by Tony Atala and co-workers. And in this system, instead of using a natural scaffold like collagen, we're using a synthetic scaffold called polylactic co-glycolic acid. So this is a polymer, which I'll describe later in a moment, but for now you can think about it like a plastic. And this plastic was cast in the shape of a balloon structure that you see here. On the inside of this balloon, we see that cells were, lining cells from the bladder were seated. And on the outside of the balloon, muscular cells that are important for bladder function, for the contraction of bladder, were seated. Together, these were cultured in vitro and then implanted in animal experiments and now in humans. And these hybrid tissues that are composed of two cell types and a polymer scaffold together promote better bladder function, better compliance in this setting. Interestingly, in this classic paper, where they first did the experiments in dogs, these hybrid bladders were also reinnervated. So they promoted nerve regeneration in the host, and these dogs regained voluntary bladder function. This was unexpected and one of those exciting areas of research that is still unexplained. So I've told you about acellular grafts and hybrid grafts. Now let's talk about completely cellular-derived grafts that have no synthetic or natural biomaterials in them. So here's an example of a biomaterial-free engineered tissue. This is a blood vessel that was made by Nicolas Leroux and colleagues. And in this system, they do what they call sheet-based tissue engineering. The vision is that one would take a sample from a patient, a little biopsy sample, from which one could harvest endothelial cells, which line blood vessels, and fibroblasts, those same dermal cells that I described earlier, and culture them in flasks. And if one cultures them in flasks, if one cultures the fibroblasts in flasks, one can see that the fibroblasts deposit their own extracellular matrix. They deposit the same collagen, types of collagen that you saw in the small intestinal submucosa earlier, but they deposit it on the bottom layer of the flask. And one can manipulate the culture conditions to promote lots and lots of extracellular matrix deposition. Then one can detach this system, and it creates a sheet that can be manipulated and rolled on a mandrel. This mandrel then now is a living cinnamon roll of cells and extracellular matrix, and it's cultured in this living format so that the layers of the cells and the extracellular matrix will fuse into a very strong wall. One can then seed the inside of these tubes with the endothelial cells that were originally obtained, and now you have a construct that has endothelial cells on the inside and this nice, strong surrounding structure made of fibroblasts and their extracellular matrix. And these can be implanted in patients. And these are what these tissue-engineered blood vessels look like. And these blood vessels have no scaffolding material that was introduced from the outside. Okay, so these are the different flavors of engineered tissues that exist. If one were thinking about designing a bioengineered tissue to replace a particular organ function, one important thing to note is that as tissue engineers, we can frequently, we are unable to replace all of the functions of interest. So, for example, if one thinks about the functions of skin that are being replaced, it's simply the barrier function in the example I showed you. Those pieces of skin have no hair follicles, no sweat glands, no immune cells, and that's a deliberate choice on the part of the tissue engineer to go after the function that's most critical for life. Similarly, if one thinks about pancreatic tissue engineering, one often sees that folks are interested in the implantation of beta cells that will produce insulin in a glucose-responsive way for patients with diabetes. There are many other functions of the pancreas that are often excluded, therefore, from tissue engineering. And again, this is a deliberate decision on the part of the tissue engineer. So it's important to keep in mind that one can't do everything. One has to have priorities. Okay, well then, once you've made your decision about what functions you're most interested in, what would one then do to go further and actually engineer a tissue from scratch? So the next thing to do would be picking some raw ingredients. The raw ingredients that one has to work with are cells. There's actually a variety of cell sources that one can consider. So there are somatic cells and stem cells. Somatic cells are cells of the body that are not germ cells, that is, not eggs or sperm. So they're part of the adult organism. And they can come from the person that you plan to treat. Those are called autologous. They could come from another person, that would be allogeneic, but the same species. Or they could come from an organism that was another species, and those would be called xenogeneic. And in tissue engineering, one actually sees examples of all of these types of cell sourcing. So for example, the skin that I showed you earlier was allogeneic. The dermal fibroblasts and the epidermal cells were from a non-self human. to create those skin tissues. Another source of cells, which is becoming more and more exciting, is stem cells. One can think about using both adult stem cells or pluripotent stem cells that have been derived from various methods that I'll describe in a moment. So one certainly needs raw ingredients of cells. The other thing one needs is biomaterials. And finally, one needs to culture these together in a way that the cells have sufficient nutrients to remodel those biomaterials and create a tissue. Once one has the right raw ingredients, we have to think about fabrication. How to assemble these structures, how to process these, how to create culture environments, how to preserve them so that they can get to the clinic, and finally, how they can then integrate with the host at that site. So in the next few slides, I'm going to talk to you about some of the issues that are related to both the raw ingredients and the fabrication of engineered tissues. First, let's talk about cells. So if one is thinking about somatic cells, culturing primary cells, they can be derived in various ways, from an adult organism, from an embryo, or from an egg. Regardless of the source, what's typically done is these are mechanically dissected in the laboratory, and then they can be further processed in one of three ways. If they are just slightly processed, and they retain their three-dimensional structure from the in vivo environment, we call that organ culture. These organ cultures are often done at air-water interfaces that mimic the environment of particular tissues in vivo. For example, the skin that I showed you earlier is actually cultured at the air-liquid interface, just like the skin on your body is at an air-liquid interface. One could further dissect these tissues, finely chop them, and culture them in a flask, and allow cells to grow out of these structures. That's called explant culture. And that's actually the way that the bladder cells were first derived that I showed you earlier. Finally, and more commonly now, these tissues can be enzymatically digested and create a single cell suspension, a suspension of cells in a liquid. And those can be cultured in flasks, and if they are a proliferative cell, if they can grow, once they grow to what we call confluence, once they fill up that flask, one can remove them, put them in a new flask, and passage them, that's called passaging, grow them, expand them over and over again. And that's how you would create the cells for your engineered tissues. One limitation of using somatic cells was first described by a scientist named Hayflick in 1965, and that's known as the Hayflick limit. So what he observed in his experiments was the following. If one takes fibroblasts, those same fibroblasts that I was describing to you earlier, from patients of different ages, so fetal fibroblasts all the way up into their 80s, and cultures them and passages them in the flasks, as I showed you, and counts doubling time, one sees that the number of cell doublings that one can achieve is less as the patient source is older. So as patients age, their cells are able to double a fewer number of times. And what that implies is that cells have a finite lifetime in culture, and in fact in vivo. And we know now that the molecular basis of this is the ends of chromosomes have something called telomeres, the so-called molecular clock, and every time the cell divides, those telomeres get shorter until the cells reach senescence. So this observation has led to much of the excitement in the field of tissue engineering about stem cells, because in fact stem cells don't have this property. They are what we call immortal. They elongate their telomeres every time they divide by expressing an enzyme known as telomerase. They actually are not susceptible to the Hayflick limit, and they can grow in perpetuity in culture. So as a cell source, they're a very exciting cell source. Furthermore, stem cells have been identified that are pluripotent. So here's a diagram describing a number of ways that one can make stem cells that can differentiate, that can be encouraged to grow into the various cell types of interest for any tissue of interest. The idea here is that you could have a single cell source, it could be immortal, it could be expanded, and then it could be used to engineer any tissue of interest. A further idea that's been very exciting in the field is the idea that one could make individualized pluripotent stem cells. This is the idea of patient-specific or personalized medicine. So let me walk you through how this idea goes. There are a variety of ways to make these cells. So in this system, what we see is that you take, again, a fibroblast, a skin fibroblast, just like the ones I showed you earlier, and they have 2 copies of DNA. So these are somatic cells. And one can take out the nucleus of that cell and put it in an enucleated egg, okay? Those cells then will start to grow into a blastocyst structure, shown here. And the blastocyst, which is the early embryo, has a pocket of cells known as the inner cell mass. If one cultures cells from the inner cell mass, one can create so-called pluripotent stem cells or embryonic stem cells. And the pluripotent stem cells, the embryonic stem cells, will have the same genetic identity as the original patient. Another way to create pluripotent stem cells from that patient would be to take the same somatic cell that we saw earlier and fuse it with an embryonic stem cell. And that process of fusion also generates patient-specific pluripotent stem cells. Finally, recently, in a very exciting set of discoveries, Yamanaka and coworkers have defined a way to reprogram somatic cells without going through an ES cell intermediate or an egg intermediate. And what they did in their classic experiments was show that a certain number of molecules known as transcription factors could be expressed in a somatic cell, and that transduced cell would be reprogrammed into a pluripotent stem cell. So this now offers the potential to create differentiated cells that are genetically matched to patients. It offers the further possibility to make pluripotent stem cells from patients to fix a genetic defect in the laboratory, and then to create a tissue out of those genetically altered cells to give back to the patient. So given the Hayflick limit, the promise of stem cells has been very exciting in the field of tissue engineering. Okay, so we've talked about cellular ingredients. Now let's turn to biomaterials. So one classical tenet of biomaterials in the tissue engineering field is that one wants to match the degradation of the biomaterial with the synthesis of extracellular matrix scaffolding in the host. So one would not want the biomaterial to degrade too quickly, otherwise it wouldn't perform its scaffolding function. On the other hand, one typically would not want the biomaterial to be persistent indefinitely, because one might incur what's called the foreign body response. So where does this concept come from? This concept was originally defined by Janas and coworkers in these classic experiments that were done in the skin. In these experiments, what they did was they made tissue engineered skin that had a variety of degradation rates. And what they looked at was the wound healing time of a full thickness wound, a cut through both the epidermal and dermal layers of the skin in an animal model. And what they found was that if one didn't put any scaffolding in that wound, it would contract and close very quickly and form scar. And that was what we were hoping to prevent with the use of the tissue engineered scaffold. If one then implants a tissue engineered scaffold that has a very rapid degradation time, those wounds proceed to contract just as in the control conditions, as if there was no scaffold in the system at all. However, if one further cross-links the material, one can see that one can delay that wound contraction time, as seen here. And then after that, if one makes the material even less degradable, there's really no benefit to that, and the wound actually just persists the same amount of time. Okay, so these experiments led to the idea that there's an optimal degradation rate for tissue engineered scaffolds. And one can actually see that in the CIS experiments, in this bottom graph here, they've conceptualized this as follows. So the degradation time of the material is decreasing, and the production of extracellular matrix is increasing. And the CIS actually bridges the gap in that in those two time scales. So it's important to match the degradation with the host synthesis. So that's a material property that's important. What are the materials actually? So there are a variety of synthetic scaffolds that one has to choose from. And if one looks in the community, we see new chemistries coming out on a daily basis. I've just listed for you here some of the classical chemistries that are used for tissue engineering. And many of them are what we call polymers. So for example, PLGA is polylactic co-glycolic acid. These are macromolecules that have repeating structures, in this case of lactic acid and glycolic acid. And when they're implanted and they degrade in the body by hydrolysis, by the interaction with water, they degrade into units of lactic acid and glycolic acid, which already occur in your body. So these are actually originally what sutures were made of, biodegradable stitches. So there are many, many novel polymeric materials that have been developed. Another very popular one, which has the property of being a hydrogel. Again, a hydrogel is a material that has absorbs a great amount of water and has this sort of intuitive feel of gelatin. Here we have a polyethylene glycol, which is a very popular biomaterial these days. And you can see that it looks a lot like a soft contact lens. What's interesting about some of these hydrogel materials is that rather than being pieces of plastic, where cells can be attached to the plastic and they can remodel the plastic, these are materials where cells are embedded within the plastic. And this material in particular is very interesting because it can be cross-linked with light. So the way the experiment works is you take polyethylene glycol macromers, so long chains of polyethylene glycol, you mix in cells and a chemical that's sensitive to light, and when you shine light, you get a network of polymer that looks like this contact lens structure with cells embedded within it. So these are examples of synthetic scaffolds, and one can tune these to have different material properties. They can be inert, like polyethylene glycol. You can modify their chemistry to interact specifically with cells. For example, one can put in peptides that would bind to cellular receptors like integrins. They can have different mechanical properties, which we know is important for some cell types, and then they can have different mechanisms of degradation. So they don't all degrade just by interactions with water. Some of them can degrade in very specific ways, for example, by interacting with enzymes that the cells are making. Another example of a kind of scaffold that one would use are the so-called natural scaffolds. So I already mentioned collagen earlier. Collagen is one of the most popular natural scaffolds. Hyaluronic acid is another natural scaffold. The cis material that I showed you is a more complex natural scaffold because it contains all the extracellular matrix molecules that were in the submucosa, in addition to the growth factors and other bioactive molecules that would be in that natural material. An example of such complex natural scaffolds that is becoming quite popular these days is an idea that was first described by Doris Taylor's group a couple of years ago. And these are whole organs that have been decellularized in much the same way that the small intestinal submucosa was decellularized. So here what you're looking at is a picture of a heart that's being perfused with a detergent. And you can literally see the cells melting away so that you have what they call a ghost heart at the end. This preserves all of the microarchitecture of the organ that I described to you earlier. You can see here under the microscope that it has fibrillar collagen organization. And what she did in these experiments was recellularized this decellularized heart, using that as a scaffold for tissue engineering. So various groups have now gone on to do this in other organ systems, including the liver. And what's exciting about this potentially is one can think about reusing organs. Instead of having to just transplant a living organ from a donor into a patient, one can think about using cadaveric organs as complex scaffolding for cells from some of the cell sources that I described earlier. Okay, so I've described the raw ingredients of cells and biomaterials. I won't say much about nutrients except for to say that there is a whole science about how to feed cells in culture, and that's pretty well understood at this point. The next thing I'd like to just touch on briefly is how to fabricate these organs, how to assemble them, how to process them before implantation, and importantly, how to preserve them so that one can get them to the clinic at the time of need. Okay, so I mentioned earlier that tissues are organized hierarchically. One can see here in this diagram that if one starts at the level of the liver, we have this multi-scale organization where the liver acini are 1 millimeter in dimension, the liver cords here are 500 microns long, and the cells here are about 20 microns in diameter. So it's this hierarchy which tissue engineers have to try and grapple with in the sense that cells respond actually only locally to their microenvironment. So even though you're trying to build a large structure, if you care about cells performing the functions of interest, and here are some functions that one might care about: proliferation, differentiation, apoptosis, migration, or metabolism. If these are the functions of interest for having a healthy tissue, that we know that the cells are actually responding to their local microenvironment to these kinds of cues: soluble factors, interactions with other cells, extracellular matrix interactions, physical forces, and cell shape. So the challenge for the tissue engineer is to maintain the cellular microenvironment that will impact the cell behavior on the sort of 10 to 100 micron length scale, but simultaneously be scaling up this structure to be a large enough body of cells to implant to achieve a therapeutic outcome. So a good example of how cells have local microenvironmental interactions that one calls three-dimensional in the field is cartilage. We see this is a cross-section of articular cartilage, and if one looks at cartilage, one sees that the chondrocytes that make the extracellular matrix that makes up cartilage have different organizational structures. So here we can see that they are in these columnar structures, whereas in the superficial layer of cartilage that sits right next to the joint space, they actually are flat, elongated, and in clusters. So if you were to look down at the cartilage, you would see that they were in clusters. So it's been known for quite some time now that chondrocytes in culture, if one puts them on plastic of any kind for tissue engineering purposes, they will actually spread out and lose many of these functions. They will start proliferating and stop producing the extracellular matrix that one is trying to recapitulate in a tissue engineered construct. And so in fact, if one embeds them in a three-dimensional microenvironment where they have this round shape and more favorable mechanical properties, one stimulates them as they are stimulated mechanically in vivo, one can actually improve the quality of the extracellular matrix that they produce and therefore the mechanical strength of the graft. So this is just one example of a tissue where the three-dimensional organization of the microenvironment, in addition to the chemical composition of the microenvironment, is very important. And one will hear a lot about 3D tissue engineering in years to come. One can then think about how to assemble these microenvironments where cells have very precise local chemical and mechanical cues into larger structures. There's a set of technologies emerging known as 3D fabrication technologies that have come out of the rapid prototyping field that are starting to be borrowed in tissue engineering. Some people call this organ printing. This is an example of a couple of parts that were made in a layer by layer fashion known as stereolithography. And stereolithography is one example of one of these rapid prototyping technologies. So in this technology is what's done is a three-dimensional drawing is drawn on the computer and sent to this robot. And the robot has a movable stage that sits in a vat of light-sensitive material. And in every layer of the material, light is shown in a pattern. It polymerizes, and then the stage drops, and the next layer is built, and so on and so forth. And using this, one can create complex structures, some that are actually biomaterial structures. Some folks envision that one could make personalized tissue engineered parts using a technology like this, because one can envision going from three-dimensional medical imaging data like CAT scan data to tissue engineered parts. Another thing that's required is to culture these tissues before they're implanted. And these are a couple of examples of the kinds of bioreactors that are designed and used in the field. At the top, what you see is a quite well-known bioreactor developed by NASA. This is a circulating bioreactor. and it never allows the cells to touch the walls of the bioreactor, so they're forced to aggregate in three-dimensional clusters. And this has been shown to promote tissue formation in both cartilage and heart cells. Furthermore, techniques from the computer chip industry are now being borrowed to create microreactors. So here what you're looking at is a cover slip and some inlet ports from a microfluidic device. And if one looks in the microscope inside that cover slip, one can see these little tissue units that have microstructure that have been fabricated in the computer lab at 100 micron length scale. So they have three-dimensional microenvironments that have been specified in a scalable fashion for building larger pieces of tissue. So one can assemble tissues, one can culture these to mature them in the laboratory, and then finally one needs to think about how to preserve them to deliver them to the patients. So there's a whole set of technologies that have grown up in the field about cryopreservation. I should say that not all tissues are frozen. Many are actually just refrigerated to 4 degrees C, and in fact the skin tissue engineered product that I showed you earlier is sent that way to the clinic. However, in the field of cryopreservation, which most of us agree will be important to really make this technology have its highest impact, there has been a lot of research on how best to make cells, especially in three-dimensional structures, survive the insults of cryopreservation. So the insults of cryopreservation have now been categorized into 2 classes of insults, and those are depicted here. So I sort of need to walk you through what a cell feels as it gets frozen so you can see what the 2 classes of insults are. So if you have a cell here and it's been cooled just under the seeding point of ice, and one artificially seeds external ice in the environment, 2 things can happen. If one slowly cools the solution further, you can see that water has time to cross the cell membrane and leave the cell and form ice in the extracellular space. And this causes what people call dehydration injury to that cell. You can see that its shape has shrunken, there's a concentration of the intracellular solutes, and those cells, when they are thawed again, exhibit damage from this dehydration process. If instead one takes this solution and rapidly cools it, one actually finds that you can have intracellular ice crystal formation. The formation of intracellular ice crystals in and of itself can cause damage to cells. So the process by which cells are susceptible to slow cooling or fast cooling turns out to be cell type dependent and dependent on various additives in the system, as well as the three-dimensional architecture of the tissue. And so this is being worked out now for many, many cell types in the field. Okay, so to conclude, I've told you that engineered tissue replacements can be made from combinations of cells and biomaterials to replace a subset of tissue functions. I've told you that the cells can be derived from somatic cells or stem cells. I've told you that the biomaterials in these tissue replacements can be natural or synthetic. I've also told you that tissue structure in the body is hierarchical, and therefore the tissue engineer has the challenge of maintaining the cellular microenvironment in a way that can be scaled up to build a tissue that's large enough for therapeutic impact. And finally, I hope you've noticed that the convergence of cell biology, medicine, and engineering is really advancing this field. In part 2 of my seminar, I'll be telling you about our work on liver tissue engineering, both in the progress we're making towards therapeutic goals, like the ones I've described, as well as progress that we're making in using these engineered liver tissues to advance scientific discovery.